Now we're going to turn to our final and last panel session of the day. This is not showing up on the screen. Yeah, I'll let you look at that while I introduce the moderator, which is Dr. Carol Barnes. Dr. Carol Barnes is a Regents Professor in the Departments of Psychology, Neurology, and Neuroscience. She is also the Evelyn F. McKnight Endowed Chair for Learning and Memory in Aging and the Director of the Evelyn F. McKnight Brain Institute at the University of Arizona. I am so delighted that she could be with us today and moderate our third session on improving the health span. Please welcome Carol to the stage, and we'll get these slides figured out so you can see them.
Good, thank you. Super. Thanks everybody for, inviting me today. And, all right. There we go. Okay, good. There I am. Before I introduce the first speakers in this series on the session of Improving Healthspan, I want to take this opportunity to tell you about two efforts at the University of Arizona that my colleagues in Tucson and across the country are involved in that fit squarely, I think, under the theme of efforts to improve brain and cognitive health span of individuals. The first is the Evelyn F. McKnight Brain Institute. I'm the director of this institute, and Lee Ryan is the head of psychology, but she's the associate director of the institute. We are one of 4 McKnight Institutes across the United States, and the Tucson Institute was established in 2006. We were the youngest of the institutes, but vibrant. The research mission of the Evelyn F. McKnight Brain Institute is to better understand brain changes that occur during normal aging that lead to memory changes and other cognitive function across the lifespan. So the goal really is to understand these brain changes well enough so that we can eventually develop practical lifestyle recommendations and treatments that will improve the quality of life for older individuals. So top researchers across the University of Arizona have joined the Institute, working in fields as diverse as psychology, neuroscience, molecular and cellular biology, physiology, neurology, optical sciences, biomedical engineering, and others. The other McKnight Brain Institutes are in Birmingham, Gainesville, and Miami. And we are all working together across these McKnight Institutes to explore the brain and to discover how age-related brain changes affect cognition later in life. The next project that I'd like to tell you about is really a very unique project centered at the University of Arizona in Tucson that we call the Precision Aging Network, or PAN for short. I'll devolve into PAN probably as we go along. And I'm the principal investigator of PAN, but Lee Ryan, again in the audience, is on the executive committee for PAN, and she is also the lead of one of 4 of our projects in that grant. Also here at the meeting are 2 other participants in the PAN, is Esther Sternberg, who's lead of the Non-Invasive Technology Core, and Ray Runyon, who is the co-lead of that core. We were awarded a $60 million grant from the National Institutes on Health to launch this precision aging network in September 2021. And the primary goal, of course, of the Precision Aging Network is to close the gap between human lifespan and cognitive health span. We aim to achieve this goal by developing the essential scientific knowledge and appropriate technologies for the application of precision medicine concepts to predict individual brain health risks and to develop personalized solutions. We're talking about individual differences here. It's so important across all of what we do. Our perspective takes into account a person's genetics, lifestyle, environment, other factors to customize guidelines for healthy brain aging. We use interdisciplinary methods that can examine brain and cognitive health in large groups of people. Right now, one of our projects has over 380,000 participants, as well as more targeted in-depth studies of individuals. This is the project that Lee Ryan heads, to target about 1,600 individuals across 4 locations in the country: Tucson, Miami, Baltimore, and Atlanta. To give you some idea of how spread out we are across the university, we have faculty from 5 different colleges participating who are affiliated with 13 different departments and 8 different centers and institutes around campus. In Phoenix, we also work with scientists at the Translational Genomics Institute and the Arizona State University. And then we have collaborators across the United States working at 6 additional research universities: the University of Miami, Johns Hopkins University, Emory University, Georgia Tech, Georgia State University, and Baylor College of Medicine. Now, across the projects in this grant, we ask questions such as, what factors might impact healthy brain function as we age? We also ask, can we predict, prevent, or slow the decline of brain aging on an individual basis? So together, the investigators in PAN are very ambitious. We hope to shape the future of cognitive health in older individuals across the country. And one final request to participants here that have been with us today is for you to consider going to the entry point for the Precision Aging Network, the MindCrowd website, which you see here, to take the memory test and answer some demographic questions. It only takes about 10 minutes, and the only requirement is that you're above 18 years of age. So just type in mindcrowd.org and become a part of this large project. And if you have any further questions, either Lee or I could answer them for you, or Esther or Ray could help answer them. And so now I turn from the couple projects that I had the opportunity to tell you about that we're doing at the University of Arizona, but I want to go on to our speakers for today. And our first speaker is Dr. Janko Nikolic-Zugic, who is the Bowman Professor and head of the Department of Immunobiology at the University of Arizona. He is also the co-director of the Center on Aging and a fellow of the Gerontological Society of America and of the American Aging Federation. His research interests are in the basic mechanisms of immunity and how these mechanisms deteriorate with age, with an emphasis on how to eventually ameliorate immune dysfunction with age, which would provide, of course, Healthspan benefits, the very topic of this whole meeting. So his talk today concerns a project that aims to identify the defects in anti-tumor immunity with age in a specific form of skin cancer, which is hard to say, but I'm going to say it: cutaneous squamous cell carcinoma. And while it's known that T cells play a role in destroying this type of cancer cell, it's not fully understood how aging T cells fail in tumor response to these cells. And so the aim of the experiments that he's going to talk to you about today is to identify what these age-related defects are in the T cell mediated anti-tumor responses. And what he hopes, and what I hope, is that this is going to allow him to devise and test treatments to reduce the burden of skin cancer and promote healthy aging. Again, the theme of this conference today. And his discussant today will be his I think, NIA program officer, Dr. Mulu Tilahum. He directs the program on immunobiology. Mulu, can you help me? And immunobiology. I didn't get a chance to get more information from him today. I just met him a few minutes ago. I'm sorry that I didn't give you more airtime here. Now, our second speaker for the day, I'm going to introduce everybody and then we'll go back and forth just to give you the overview. Oh dear.
Yeah, okay, that'll just take you through the slides, but go ahead.
All right. So our second speaker for today is Dr. Anita Kilar. And Dr. Kilar is an assistant professor in the Department of Speech and Language Hearing Sciences and the director of the Language and Neuroimaging Laboratory at the University of Arizona. And Dr. Kilar's research are in the area of cognitive neuroscience of language and neurogenic communication disorders. Specifically, her work examines cognitive and neural factors that affect language functions, how they change across the lifespan, and how they interact with stroke or other neurodegenerative diseases. So in her talk today, she'll focus on an atypical form of Alzheimer's disease that impacts language function before it impacts memory, and that this form of AD first impacts the function of the I think is the left parieto-temporal region, causing a form of primary progressive aphasia that's called, okay, here's another one, called the logopenic variant. Did I do it right? Okay. So it's been shown that behavioral interventions can be successful in ameliorating the loss of some of the language components or language functions in this particular variant of AD. So the experiments that Dr. Kilar will talk about combine these behavioral interventions with non-invasive transcranial direct current stimulation. That is fMRI, that's MRI guided. Guided in the sense that she wants to do the stimulation in the intact tissue, still remaining. And the prediction is that by stimulating intact tissue involved in language that's been selected based on these scans, along with the behavioral treatment, this will produce a more impactful and beneficial effect than just the behavioral therapy alone. So, and the discussant for, oh geez, you know, this is crazy, I know. Okay, so the discussant is right here for Dr. Kilar's talk is Tabassum Majid, who is a program officer in the Healthcare Delivery and Disparities Research Program at the Patient Centers Outcomes Research Institute. And she has a long history, a long interest in neurodegenerative disorders in the aged and in health services and social programs tailored to the needs of both caregivers and care recipients, okay? Okay, so now I think we can go back to Dr. Nicholas Zugic's talk, and all righty?
No problem.
You can get there.
Yeah.
Okay, and the title of his talk,
Would you
he
prefer to
doesn't
start
have
on
it.
this slide or the
That's
next
all right, yeah.
I'm looking.
The title of his talk is, here's Janko. It's Janko. Thank you, Janko. Appreciate it.
Thank you so much, Carol. I'll talk about something as you said. So as Carol said, one of my important hats is the co-director of the Center on Aging, and so the awe is being a co-director with Mindy. So and the only thing that I will mention along those lines is that our center is the only one in the state and one of the very few in the nation that has the missions of patient care, research, both fundamental and very applied, outreach, and education, as well as the cross-state mission by the Board of Regents. So the talk that I'm going to give today actually is the credit to Karen Hastings a lot more than myself. Karen and I are doing this together, and Karen is, I think, now full professor at our College of Medicine, Phoenix. She's a clinical dermatologist as well as a great basic scientist, and has been studying these problems that we will talk about a lot, except for the aging part, which is where I come in. So the other important point to mention is that it's going to be very, very mouse-centric. And the reason is that we typically need to understand these things in terms of how they fundamentally work before we jump into humans. However, when it comes to relevance of this, the entire concept of checkpoint inhibition has arisen entirely in mice before it was translated into humans due to the work of Jim Allison and others. And so the proof of principle here holds pretty tightly. So this is just the incidence of cancers, with aging. That would not surprise anyone. You can see how that goes up. And it is also true for various individual cancers. Whichever way you plot them, they go up with aging. So they also do share common molecular pathways, and that stands at the basis of the concept of geroscience, which posits that the fundamental building blocks of aging are universal and likely activated in many chronic diseases. And that for this reason, if we can understand the fundamental posits of the aging process, we may be able to actually prevent or delay many of the chronic diseases as well. So in this case, we're talking about increased mutational load, telomere dysfunction, DNA damage, immune dysregulation, increased inflammation, and so on and so forth. So then when we think about how this will play out in the case of the tumorigenesis, aging will be everywhere here, but there will be metabolic issues and concepts that underlie this, epigenetics, immune surveillance, all of that leading to tumor progression or tumor stoppage if we're successful at all of this. And here you can see how this plays out in the case of the breast cancer, where you will have the initial changes with transformation and microenvironment remodeling leading to either in situ lesions, primary tumor that then breaks through the barrier, and the metastatic forms of cancer. Sadly, immunity and cancer in aging have not been really studied very well, and so this is where we have a major gap in knowledge. Part of the reason is that the mechanisms to get NIA centers that have different foci together on a single point have been sometimes somewhat difficult to enforce. If there's a lot of will on both sides, yes, that's fine. But unfortunately, in many cases, very often when you write a grant that straddles those types of things, the study section will tell you, oh, this is great, we're a cancer study section, this is all perfect. Just take that aging stuff out and we may fund you, and so forth. And there's been thanks to Felipe Sierra, thanks to Ron Kochanski and other people in Mulu and other folks at the NCI, now we're getting to the point where this is something that is being overcome slowly, and we're getting to the point where I think we're going to be able to circumvent these barriers. Let's talk about the cancer that we're trying to address here, the cutaneous squamous cell carcinoma. It's a non-melanoma skin cancer, and non-melanoma skin cancers, which include this cancer, are the most common cancers. We have more than 1 million cases per year in the Medicare population alone. Majority of the primary tumors can be treated very efficiently by excision, and they're completely cured by excision, but advanced tumors that are not resectable do cause significant mortality and morbidity, and about 4% of those develop metastasis, and about 2% will die from metastatic disease. If you multiply that by the huge frequency of about 1 million per year, now you get actually to the point where this actually results in similar number of deaths each year as melanoma, which is a far more aggressive cancer, but doesn't occur that frequently. And this is particularly the case in southern US states, such as Arizona. What do we know about the control of this cancer? Well, we do know that T cells are very important. We know that immune checkpoint inhibitors, which work on T cells, particularly those with anti-PD-1 monoclonal antibodies, improve overall survival in advanced cancer, and that about 32 to 46% of advanced patients respond to this therapy. Solid organ transplant folks that are on immunosuppression have hugely increased incidence of this cancer and hugely increased risk, between 65 and 235-fold higher risk. That is very similar to what we see as a general deterioration of immunity with cancer when it comes to infectious diseases. And just to give you a good relevant stat, over the course of the SARS coronavirus pandemic, people that were 80 years and older died at 270-fold higher clip than people between 18 and 39 years of age. That is a phenomenally huge vulnerability. It's not 270%, it's 270-fold. So this basically mandates that we understand what's going on here and that we figure out how to improve immunity. So what we know also when it comes to older population is that this incidence of these skin cancers is highest amongst people 75 and older, 208 per 100,000 people. Median age of folks in clinical trials is between 72 and 79 years of age. Immunotherapy is showing efficacy, but we really have no idea how aging is impacting the efficacy of this. Analysis of the response to immune checkpoint inhibitors with age has so far given conflicting results in other cancer types, so we just don't understand this whole thing very, very much. When we're thinking about how are we going to start studying something like this, as I said, immune checkpoints have been defined in mice. This is something that we know and understand. We have good models. Our lab has shown that the consequence of poor T cell responses in aging to infection come because we have too few of the naive T cells that are supposed to respond. We've lost about 2/3 of them, if not more. And also that they do not do very well in the site where they're supposed to do their business. Most specifically, the lymph nodes where the naive T cells are supposed to be initially primed and initially see their microbe that they're reacting to are disorganized and very much affected by the process of aging, which is one of the barriers that has been underappreciated so far. So then we need to figure out how to fix all of these things, but we first of all need to figure out, is this also still true in age response to cancer? So what agents, or what tumors shall we use and so forth? Well, we like to use these B6 mice that are, of course, somewhat abnormal themselves because they're a simplified model of the immune response. They have relatively few MHC molecules. They're, of course, inbred. This is the model that we have used. We've injected them with the melanoma here, which is a first proxy to see can we observe the same things that we normally observe in the response to infectious diseases here. And then we would look at tumor progression and we would look at what cells are coming inside the tumor, what T cells are coming inside the tumor to try to deal with it, to destroy it. And so what we have done here, these are the age groups that we have studied, and the B6 mice start to draw social security at around 18 months of age. That is where we consider them somewhat older. But of course, there is this issue that you've heard from Carol, that they do show a huge heterogeneity. You really would love to study them at 24, 26 months of age. That's somewhat difficult, sometimes impractical, sometimes impossible. So we started these different ages to check them out. And so the first surprise that we found was that older mice were not only good, but actually better at rejecting this particular tumor. So you can see here that the most sensitive group was the one at 3 months, and that the mice at 22 months did really well, and that their tumors were pretty small. Now, this is a pretty immunogenic tumor. This is not a native tumor. We actually plopped in another foreign molecule in it to target the T cells to this tumor and to be able to study them. And that molecule is chicken of albumin. Don't ask me why we do this, but we know a lot about this response in general. It's a good model. So maybe that was the reason why this didn't work. And what we found out is that when we looked at the infiltrating cells here, you see that these cells here in 18 months old mice actually expressed a lot of things that would kill the tumor, like tumor necrosis factor and interferon gamma and granzyme B, which makes the T cells kill their targets and so forth. So all of this was consistent, and not only that, but the cells that express molecules that you could inhibit to get the tumor immunity better were very infrequent in old mice in the tumors. They already did not have this phenotype of partial exhaustion, which is what we're trying to remedy with checkpoint inhibition. And so we've repeated this in a couple of other tumor models, all of which were somewhat artificial, and we've repeated and seen the same result, essentially. So we've seen that the lymphoma and a mastocytoma were similarly rejected. These were all, again, very highly immunogenic tumors. So one of the possibilities here is that, we're giving the mice something that they will react super strongly to, and there are some factors that we're still trying to understand how this is all working. But at that point, we've basically turned to Karen and told her, Karen, this is really not working. We need a real model of squamous cell carcinoma. Let's get something that will be closer. And so what Karen has done is she has actually used UV light, solar light, to actually generate the real squamous cell carcinoma in mice. She used a different strain of mice, BALB-C, which are known to have weaker cellular immunity. B6 mice actually love to make good T cells, and so this could have also worked against us. But Karen has done these different types of regimens, and the most important one was that she used the real solar UV light. And when she did that, she could generate these lesions in the skin of mice. She could pull out these cells, culture them in the dish, make sure that she gets clonal lines, and then she showed that these clonal lines that she made can actually grow. And the first and the easiest thing is to put them in mice that have no T cells. And they could grow very, very well in these cells in these mice that have no T cells, the athymic mice. But they also grew in wild-type mice, which was very important. So that told us that we have a somewhat real model. These are all young mice. We still haven't done anything in old mice with these cells. That's going to be the next step. So she generated this one clonal cell line here, and she has done a huge amount of work to identify unique mutations and neoantigens that these mutations produce, something that we could use to track the immune response. She showed also that this particular line also cannot be contained if there are no T cells in the system. She showed that if you vaccinate with this cell line that is irradiated and therefore cannot grow, you can completely inhibit the tumor, and I'll show you some of this data in the next couple of slides. And she also showed that CD8 T cells are critical to the control of this tumor. That makes sense. They're the killer cells. They're the ones that normally we would expect to do this type of work. CD4 cells, which is the other flavor of T cells that usually modulate and control the immune response, were important, but to a much lesser degree. And she has actually then gone to identify through a whole series of complicated biochemical experiments the precise antigens, the pieces of this tumor that are changed by tumorigenesis that we can then use in the work. So one single peptide antigen that she has used can actually slow down tumor growth when used as a vaccine. So all of this gives us a great roadmap then to go into old mice and see, is this going to work and how is this going to work? This is the data of what she has done to show that this is dependent on T cells. If she doesn't treat mice with anything, which is this sort of like black line, there's, the tumor grows very slowly. If she takes away CD4 cells, it grows a little faster. If she takes away CD8 cells, it grows a lot faster. If she takes both, then it's similar to CD8s alone, a little faster over there. And so this is another type, just giving you, this is the tumor volume, and this is the number of tumors that are pretty much huge, just telling you that this depends on T cells and more on CD8 T cells than on CD4s. This is the vaccination experiment when she takes and irradiates the cell line and injects it before the tumor. Mice mount an immune response to the cell line, and now all of them are protected against this line. You can see the mice that are depicted here in violet don't develop tumors at all. If you give them just the saline, then they develop the tumors that are growing very fast. If you give them a similar cell line that was also generated by UV radiation, but is not clonally the same as this one, presumably the one that has its own different mutations, then mice are not protected. So it's telling you that this has very strict antigen specificity, which is exactly kind of what the model that we're looking for. So right now, this is where we are in the model building. We have shown that we have a new mouse model to investigate neoantigen-specific T-cell responses. They're induced by the same etiological factor as the human disease. It has the neoantigen landscape that's similar to the human disease, meaning unique to each cancer. Again, it goes back to precision cancer immunotherapies that we might very well need. But if we know that this is happening, these pipelines are now pretty quick. We can actually get this done very fast to help people. This is constrained by CD8 cells more than the CD4 cells, and vaccination completely protects from tumor challenge. Screening of a small number of neoantigens has given us a really good peptide that we can now study. And so now this is enabling mechanistic studies in T cells, and our old mouse model with this cancer is under development. So this is where we are. We're very grateful for the support, and we'd like to thank all of these wonderful people who have done this work, including folks at the ASU, and funding is here very prominently. The grant challenges have really enabled these studies in a way that we would have not either gotten together or done any of this work. So thank you very much. And I'm happy to. And so I was going to turn this over now to Dr. Tilahun, who is in the Division of Aging Biology, handling the portfolio of immunity, immune responses, and microbiome.
Okay. Good afternoon, everyone. Thank you, Yanko, and the organizing committee for inviting me to attend this very important and informative summit. I have learned a lot. As Yanko said, my name is Mulu Tilahun. I'm a program officer at the National Institute on Aging and a division of aging biology. So I manage a very broad spectrum of portfolio in immunology and microbiome. And several of the grants that I manage is Yanko's, his previous continuing portfolios, as well as the new award grants that we are going to award him very soon. So I think we held a very important workshop a couple of weeks ago that Yanko was also in attendance on advances in aging, immunity, and chronic inflammatory disease. As we age, we develop several inflammatory disease. Our immune response also declines. And one of the gaps that we identified during that workshop and discussed with the workshop attendants was how to better use lesson learned in cancer immunotherapy for enhancing healthy span or enhancing immune response in older people. So one of the presentations that Yanko just gave is one best example, and National Institute on Aging is very interested to support this kind of research. We are going to, as you know, cancer and aging is very associated. As we age, we develop so many diseases, including cancer. And there is a great need to collaborate with programs or program directors at National Cancer Institute and National Institute on Aging. And I was, last week during the American Association of Immunologists, I was discussing with several program officers that how we can, collaborate more to develop different initiatives on this area, especially on cancer immunotherapy, and how to use those approaches for immune aging, during immune aging or healthy aging. So we look forward to work with so many of you, including Dr. Nikolic, and I look forward to work and interact with so many of you. And great work, Yanko, and look forward to work with you in the many years to follow.
Thank you very much, Molo. I really, really appreciate the comments, and we're grateful to NIA for all of the support, and many of us in the room. I'd like to, just make two more quick comments. One is that Karen Hastings has already put one part of this work for funding and support through Clara Curiel's program project in cutaneous cancers, and then the two of us are looking to put all of this work then forward for other substantial funding down the line. So thank you very much with that. I think we can save some time for other presenters. And so
or some questions.
yes, that's
Do I
right.
have
Sorry.
questions from the
Oops.
audience? Yeah, we had one.
Well, if no one else, thank you. That was really wonderful, Janko. I'm thinking, of course, about our study together. We're part of the Recover study across the U.S. And what's your sense of why we're seeing individuals, in terms of their immune response, where for one individual, same virus, but one individual, they come out of that, it appears, with significant impairment to the cardiovascular system, and someone else it's to the heart, the brain, others the lungs, and even the gut. Why is that? It's not. Do you think there's different sensitivities across individuals? If that's why we see that?
There are, and I think there is the first substantial paper out of the Recover study on symptoms is going to come tomorrow morning in JAMA. And it's going to talk about 5 different clusters of symptoms that are, as part of the independent analysis that are coming out. That's telling us the same thing that you just told us in the form of a question, that, we're going to have different symptomatologies. And I think somebody has, even in that paper, they're basically described long COVID as the syndrome of syndromes. The interaction of this particular virus with different parts of our body still really remains a bit of a mystery, and we all wish that we had a more naive and a cleaner population now, so that we could exactly track the events as they happen. By now everybody has more immunity to it. So what do we know overall is that there is a level of endothelial dysfunction in some people. Why is it there? I don't really think that we know. I think that for both autoimmune predisposition, because part of long COVID is going to turn out to be autoimmune, and for the maybe coagulopathy, we're starting to look at the genetic impacts that were just not known up until now. And the simple mass of people that we're now being able to study is enormous. So it is a unique point in time where we can figure this and a whole bunch of post-infectious syndromes. How the virus allows the endothelium to be altered for such a long period of time, we have no idea, but there's no doubt that this is something that's happening as the interaction between the virus and the host. Autoimmunity, for the paper will show very clearly that everybody with autoimmune predisposition was more predisposed to PASC. So there is a risk level that has popped up there. Or maybe I have misphrased this. Not everybody, but a subset of people that go into PASC clearly has this issue. To me, one of the most relevant pieces of data have still not been published, and it's Francis Eunhyang Lee from Atlanta, from Emory, who has this really cute little assay to figure out what is your system reacting to now. So she basically takes your PBMCs, spins them down, and then lets them rest for 24 hours. And so now in serum and plasma, or actually in the supernate and what you collect off of these cells, are going to be only the antibodies that are made now, and not the ones that you have made 40 years earlier in response to childhood vaccines. So you kind of like purify that way and you answer that question. By that question, when she looked in long COVID people, almost exactly half of the people ended up not really making any antibodies at all. And the other half was making a lot of antibodies, both against the coronavirus and against the whole slew of autoantigens. So that's telling you immediately that, about half of the people are going to have some autoimmune component or some overexcitability of the immune system component. And I think that, in the same way as, there was a whole bunch of people dying of COVID, acute COVID, that we didn't understand, turned out that they had, innate immune deficiencies in type 1 interferon, either because they were making antibodies to their own type 1 interferon or because they had inability to mount the interferon response, which is essential to the early virus sensing. The virus basically made us diagnose a whole bunch of immunodeficiencies that people weren't aware of. So that's what we're talking about, but boy, do we have, a steep mountain to climb to understand it all.
Can I ask a question? So this is totally naive, and if it's too naive for you to answer, just don't.
There's no too naive.
But so this session is about, improving our health span. So is it even a hope or a glimmer in your eye or a thought that if we could find a way in early life to boost immunity, like forever, would we be just naturally getting rid of cancers? Is this an approach at all to take it back, or does immunity change over development so much that what you would need as a young adult to, fend off something would be different than an older individual?
I think there is hope, and I think that the hope is realistic. When we looked at different levels of defects, I mentioned that we lose T cells with aging, but we know how to restart the factory that makes them. There are clinically approved drugs that will regrow your thymus. They're approved for totally different indications. They're not approved for treating an older person or middle-aged person to make another wave of T cells. One of them is for stomatic keratitis in head and neck cancers that get irradiated. Another one is for prostate cancer, but they are all FDA approved, and it's known that they do this thing. But the level of defects actually is more subtle than that. There's more defects. There's a couple of more that we need to fix, but I think it's doable. And I think what you are mentioning is that the intervention point may have to be maybe between your 30s and your 40s or something like that.
Right.
And not waiting until it's, We don't know exactly what's the point of irreversibility, but we have a very strong sense that there is a point of irreversibility.
Oh, thanks.
So.
Any other questions or from the video audience? No? If not?
And just as a quick one quick comment while you get the microphone, the benefit would be not only for cancers, it would be clearly for infectious diseases that kill us a lot,
so many.
but potentially also for chronic diseases where this dysregulated immune inflammatory response plays a big role.
And I think, Tulu, you were saying that we need to cooperate across institutes and so forth, and it's really so critical,
Absolutely.
I think, to be talking to one another about this, the early development.
Well, develop different programs. For example, the workshops that we organized last week was between NIAID, National Institute of Allergy and Infectious Disease, and National Institute on Aging, because we have different interests. Similar interest that we want to support. Yeah. That's why I say NCI and National Institute on Aging should work together, especially in this area, of science that can benefit both.
Right. Thanks.
But I just wanted to clarify. So what you were saying before is really important. So it's like the Goldilocks. You want enough immune response, but you don't want over responses, and when it gets into sort of the autoimmune response. And how do you balance that? So it won't be one size fits all. It'll be some people who need it.
Yeah, I think you're absolutely right. I think that there will be people that have a predisposition towards, overly exuberant responses. And one of the reasons why we're seeing both general autoimmunity and long COVID be more prevalent in females, as well as generally the reaction to the, mRNA vaccines being more vigorous and perhaps more disabling in women, hails directly to the fact that, females have 2 copies of one of the key innate sensors that triggers that interferon response, and that senses single-stranded RNA viruses. So that's one example where you want to, be careful. Also, is the old thymus going to do its proofing in the same way, like purging the autoreactive cells as well as the young thymus? We don't know that, and in fact we have some indications that maybe it won't. So then it goes back to, when do you rejuvenate and how do you make sure that it cannot be a sledgehammer, and a lot of it may have to be individualized by profiling. So again, detailed profiling, particularly in the cases of cancer responses, but also in terms of also responses to vaccines and how we react, is going to be critically important. So I'm looking to Dean Schnellman to, partner and do a lot more of those types of things, going forwards.
Thank you, Janko.
Thank you very much.
Thank you. Okay. Okay. Are you set? Yes. Okay, Anita. Great.
Okay.
Terrific.
Hello. Thank you for inviting me here. It's really great to be here at the summit. And today I want to take this time to talk about the study which we conducted recently, looking at neuromodulation combined with behavioral therapy in language variant of Alzheimer's disease, which is logopenic variant of primary progressive aphasia. So the 2 questions that I want to ask is, I want to describe what primary progressive aphasia is, and also the second question, what can we do to improve language abilities in people who experience this syndrome. And just to bring us to real life, here are the images of people who have been diagnosed with with aphasia in general, and more recently with primary progressive aphasia. So it's a real life problem that people are experiencing. So what is primary progressive aphasia? So we can distinguish 3 diagnostic types: logopenic primary progressive aphasia, non-fluent or agrammatic, and semantic PPA. And each is associated with different neuropathology, different distribution of brain atrophy, as you can see on the slides, and different language symptoms associated with that. And it is a neurodegenerative disorder that is characterized by either frontotemporal dementia or Alzheimer's disease. And today I'm going to describe the logopenic PPA. So specifically people who are in our study, they were experiencing logopenic subtype of PPA. And the impact of this work is important because, as you know, currently there are about 6 million of Americans living with Alzheimer's disease, and by the year 2050, it's estimated that we will reach 12 million. So the population who have Alzheimer's disease and possibly experiencing progressive language disorder is increasing. What are the language symptoms in primary progressive aphasia? So in the logopenic subtypes, this is associated with accumulation of atrophy in the parietal temporal regions of the brain, as you can see on the slide here. This manifests as difficulty with word retrieval, and also specifically when we test people in the lab with repetition. But what it is really the problem, underlying deficit is difficulty with phonology, phonological processing of language, and working memory deficit, verbal working memory deficit in these people. So this can be manifested as errors in sound selection when they're trying to retrieve the word. So everyone probably had this experience that you're trying to remember a word and just like other words come up that are similar. So this is the difficulty with retrieving correct phoneme of the word and trying to put them together into a word. So it can result in something called paraphasias, for example, bank for bench, right? So they could result in a non-word or different word. And in real life, it can impair reading and writing, which are very important functional skills for people. So when people come to our lab and to the clinic, they often, this is most often complaint that they would like to read and write again. So it's, this is the goal, to translate this to functional skills. So what does phonological impairment look like? And we use several tests to do that. So I want to play a short video to show you how this works. So this one is impaired phonological manipulation of sounds or sound replacement. So this is when we ask people to replace the sound in a word and come up with a different word. So I will
say SAR. SAR. Change S to M. Okay. Say POB. POB. Change A to I. Say NUSH. NUSH. Change SH to G. Okay.
This was example of phonological manipulation, which involves sound replacement. So replacing a sound in a word. And it looks like an easy task, but it's not. Usually people who have logopenic PPA, they have difficulty with that. A different task, which is a little bit, it's a different component of phonology. It's combining words into sound. It's combining sounds into words. So here, this is what we call blending task. So in the blending task, we are combining individual sounds into individual words. And this is demonstration of this with one of our participants.
What's the word? Oily.
Oily.
Okay. Next one. E-N. Okay. T. Okay.
So in the short clip, you saw that he got pot correct, but two other words they had incorrect. He combined them in the correct way. So this is another way in which we can get a sense of people's phonological representation and ability to use phonology to form words. And this deficit translates to real life. So when we actually ask people to write single words, you will see some pattern which we call phonological agraphia. So this is pattern when participants do better on real words than non-words. So here you can see that all of the real words, even those long words, are spelled correctly, but the deficit is really seen on the non-words where they have to use phonology because they don't have the lexical support. So they have to use phonology to actually write those words. And you can see a clear deficit in a pulling phonological representations of words. And this translates to also writing, passage writing. And in one of the tasks that participants do is describing the scene. It's called Picnic Scene from Western Aphasia Battery. And they would write about the scene. It's just like free writing task. And what you can see here is the writing sample before treatment from one of our participants. In red, I indicated errors. So what you can see, there are a lot of spelling errors which impairs comprehension of those sentences. So the sentences are not well informed because of all those errors of phonology, pulling up the correct sounds of words and combining them into full lexical units. So this is what we're dealing with when people have this logopenic PPA. This underlying phonological deficit disables their reading and writing skills, and sometimes even speech. So what can we do to improve language skills in logopenic PPA? So there is this recognition that phonological deficit indeed is one of the underlying deficits, but there are not many treatments that target the specific deficit. And the treatment in stroke aphasia suggests that when we train phonology, those underlying skills, people get better on reading and writing and forming sentences. So in this study, we examine whether phonological treatment, so treating this phonological awareness skills in primary progressive aphasia combined with brain stimulation would help people. And this is Caitlin Nichols. She's a speech pathologist and she's a PhD candidate who performed the treatment with the participants. So, can we improve phonological skills in people with logopenic PPA? We wanted to see whether they can benefit from this treatment and whether it's a stronger effect when we combine it with non-invasive brain stimulation, which is transcranial recurrent stimulation. So maybe some of you are familiar with this. So during TDCS, the small current is passed between two electrodes on the brain, on the positions on the head, and some of this current will reach the brain and reduce resting potentials of the neurons. So it makes them more excitable. So when people perform a task, hopefully it will engage the task relevant regions that are involved in language. So in our study was crossover double-blind randomized design. Everyone underwent baseline assessment, and then they were randomized to receive either TDCS first or sham second, or sham first or TDCS second. And we have two phases of treatment. Each treatment phase was 2 weeks with 10 treatment sessions every day. So they received 2 weeks of treatment in each phase. Here are some demographic characteristics of our participants. The age was about 70 years old. They were about 18 years of education on average, and time post-onset was from 1 year to about 5 years post-onset. And to determine where to place the electrodes on which areas of the skull, over which brain areas, we perform both structural and functional imaging, and to determine the areas that are responsive to language and also areas which are not atrophied. So we placed electrodes over areas that were responsive to language task. So this was done individually for each person. In addition, before the session, for each participant, we perform current modeling, so to determine whether to confirm the current flow and direction of current flow on the head, and then we positioned electrode over that area. So I wanted to, don't know if I have time to show you this, but I wanted to show you short clip of of the session with the participants, and they wearing masks because we did it during COVID. So everyone, most of participants wore masks. So the treatment targeted phonological manipulation skills. So we use those tasks such as blending, deletion, and replacement during the treatment to eventually they could come up with the correct lexical item. I have only 5 minutes, so I'm going to go to the results. Yeah, okay. It's not very long. Oh, okay.
This is a little tired today. I'm going to start by saying some sounds separately, and I want you to try to put them together to make a made-up word. All right, so what we were just doing a few minutes ago. Okay, let's try this one first. Okay, mop. Mop would be the word, the made-up word. Because mop is a real word, right?
Yes.
So we're looking for mop. Okay, so you got it, mop. Go ahead and write that down. So mop. Because mop's a real word, right?
Yes. Yes.
So we want the one that's not a real word. Mop.
Mop. The P. The P.
All right. So you have mop,
Mm-hmm.
which is a real word.
So this is longer, but the whole treatment was an hour, took an hour. So we went through this process with the participant, making sure that at the end of the treatment session, they would have the correct lexical item represented. So these are the results of the study. I'm going to show group results and then few individual participants. So what we found was that on the trained tasks, we found that the group that received TDCS first, combined with behavioral therapy, they show significant improvement on those tasks such as blending, phonological manipulation, and but also you can see significant improvement on the reading task, and we didn't specifically train reading, but it's a task that uses those phonological mechanisms. So there was some generalization to this task here. Interestingly, in people who received sham first, we also saw improvement in blending tasks, but the effect was much smaller than for those who receive behavioral intervention with TDCS. And the dramatic improvements were on this writing task. So passage writing task, which we didn't train. So these are the group results. Again, a group who received TDCS first show improvement in overall writing accuracy and spelling and sentence when formed, but group who received sham first show much smaller gain, and it didn't really, there was no maintenance to follow up. But the group who received TDCS, we saw very strong maintenance at the follow-up, and also they maintained this above the baseline. And I want to also show individual responses to the writing task. So this is a pre-treatment response for the participant you saw earlier. You can see that there are some spelling errors, a number of sentences of well form is decreased. This is post-treatment, so this is dramatic improvement of the writing performance post-treatment. We can see that there are sentences well formed, there are reduction in spelling errors, and this is also showed up in the formal analysis where we see significant improvements in the TDCS phase, which is maintained over the sham phase and to the follow-up. And the participant with similar, with the same task but slightly different profile, here again, we see a lot of errors at the pre-treatment and then improvement at post-treatment with number of sentences well formed increasing. And again, strong effect in the TDCS phase and maintenance to the follow-up phase, which you have to remember, it's a progressive disorder, so we're really excited about this maintenance, that they can maintain, and study takes 6 months to complete. So they maintain over all this time. So can we improve language abilities in progressive aphasia? Seems like yes, we can do that. And we also show that there is generalization to writing. So it's a functional skill that participants are often have difficulty with. What we see, which we want to investigate a little bit more, is that people who receive the TDCS first show greater benefit, even over the waiting period and then in the follow-up. So we want to investigate that a little bit more to understand why, what is happening there. And in the future, I would like to extend this to other subtypes of PPA, non-fluent and semantic, to design specific treatment protocols for these variants. And my long-term goal is to transition this to clinical practice, so actually patients and families can have benefit of this. And I want to play as the last thing testimonial from one of our participants who she completed the whole protocol, 6 months of intervention. So here we go.
And do you feel like it's helped you?
Oh my goodness. Yes. I don't know how people do it who don't have you. It's obviously changed my life.
Oh.
No, really. And I'm very grateful for everything that you have done for me.
And do you feel like
So I feel like this has real impact on people's life. It's not only in the lab, but also in the real world settings. So I would be really excited to translate this to the clinic. And this is my research team and collaborators, and I couldn't have done this without them, so thank you to everyone. And thank you for giving me this grant to work on this project. Thank you so much.
Okay, next we'll hear from Dr. Majid.
Well, thank you so much, Dr. Kilar. I think my first duty is actually to help discuss some of the work. So if you join me, first of all, I was so excited to get to review your paper and see it, because from PCORI's perspective, we're really the application-based funder, that works in tandem often with the NIA, and it's such a deep interest of ours to see things translated into practice. So that's really kudos to you. I had 2 questions for you, and maybe then can pose some challenges and conversations from the audience. So just want to look at my guide here. Do I have 2 minutes to do this part, and then we can do the slides? Is that okay? Okay, great. So the first question is, can you talk a little bit about how the caregivers or families were involved in this process? I know you didn't have time to get into it, but I'm sure they came with individuals. I'm sure you saw beautiful interactions, and how would you think about that?
Yes, so it's really important because people with primary progressive aphasia need support from their family. Some of them are functional enough that they could come into the lab themselves, and there were few participants, but this is a really important point that the support from family is essential. So we always met with the family and with the patient before the study. We explained the study so that they understand, provided resources. So we always try to be really family-centered when we did that with participants. So not only the person who participates, but also caregivers understand what's going on. And we collected also measures from, I didn't present this, but from family members and from participants at each stage of the study to make sure what is their perception, how do they feel, do they feel that they're improving, or do they feel like this is helping? So we have all those measures as well. Yeah, so really important
awesome.
question.
Thank you so much. And then the other question I had for you was, how did participants react to something that maybe as scientists we would describe it as non-invasive, but when you look at kind of a picture from their perspective and you're seeing kind of things attached to them, you're kind of seeing the clinical look, for lack of a better word, of the treatment. I mean, how do you think about that in terms of scalability and from the patient experience standpoint?
Yes, that's true. This is really good question as well. So we, again, we met with participant. We described what we're going to do in detail. We created a video tutorial so they can see how the session is proceeding and that they have to wear the electrodes. I always met with them before the study and explained how it's going to work. I applied this to myself a few times, so I know. So they often experience tingling sensation or itching sensation. So we measured all that. We ask every day how they're feeling and what they are experiencing. And the most, no one really experienced any type of severe side effect. The most was itching that they described. So itching under the electrode. Yeah, so they tolerated that quite well, the stimulation.
That's awesome. Thank you so much. Maybe we can take some questions and then I can kind of continue into the discussion part. Any questions? Is that okay? Or should I do something different?
I think you should just go ahead with your discussion part, and
Sounds
then we'll
good.
have questions to
At
you
the
and
end, to both of us
yeah,
at the
to
end?
both of you.
Okay,
Is that all right?
fantastic.
Okay.
Okay, wonderful. I don't know. Do you want to stay? You're welcome to stay. That's okay. I'm just not sure. Yes. Okay, fantastic. Yeah, so I think one of the pieces that we wanted to share with you today is sort of the different types of funding that are available that may be applicable to many of your work. And I know specifically applicable to Dr. Keelar's work, given that you're already working in human beings and you're thinking about the application. Sounds like you really took the patient and caregiver perspective to heart. And one of the things that we do at PCORI is think about this from a variety of stakeholder perspectives. So I'm just going to go through briefly who we are, what we fund, why we're interested in older adults, and then I assume we're going to take questions at the end. So we are really an independent research institute that was authorized by Congress in 2010 as part of the ACA. And so we fund what is called comparative clinical effectiveness research, which is CER, and the fundamental principle behind this research is really to help individuals and other stakeholders decide what the best option is, whether it be a treatment, whether it be an intervention, between two or more choices, right? So that means that everyone has to be involved in the process on thinking about those individual aspects. And so I can describe that a little bit further, but essentially they're real-world pragmatic trials about what works best for whom under what circumstances. So I'll just leave the mission for you all to read at your own leisure, and I'll just keep going. So it's different in terms of not only the real-world settings that have the applicability with a lot of our CER-funded work, but we also focus on questions that are most important to patients and their families. And so in that way, the patients, caregivers, clinicians, insurers, employers, anyone else who is really interested in the answer to this work is involved in the research process from the beginning. So oftentimes as scientists and as other individuals, when we're thinking about our questions, we don't necessarily think of the end goal or experience of the individual as we're thinking of those questions. Many of us are getting better at doing that, but that's all we do at PCORI, and we insist on that throughout the process. So what does that mean? That means that we don't just do research on individuals, but we do research with them. And we make sure that they have their lived experience, which is as much of an expertise as us as scientists who have our own scientific background and our own experience. And we find, and we've measured this, that this makes research more patient-centered, relevant, and useful, and most importantly, most applicable to implementation and scale across health systems. So this is just giving you a flavor of the life cycle of a study, which you all are extremely familiar with. But really, we think about the process in which individuals can be involved at various aspects of the study life cycle, ultimately changing policy and practice in terms of disseminating and implementing the work. So there are a number of ways in which individuals can be involved. And again, we've seen results. So we know that health systems now are formulating policies on promoting patient engagement in research. We know that journals are now reporting reviews, and also that universities are also offering seed money, much like some of the centers that were described earlier on research-patient partnerships. So how much have we invested in the area of older adults? About $300 million so far. with 52 studies focused on older adults, and this number's growing. We also have a number of populations and conditions that we've focused this work on. And I, like my other program officer who spoke earlier from the NIA, both manage and co-lead several strategic initiatives and portfolio of projects in this area. So happy to talk about any of them at a later point. And we also have engagement studies on older adults, which is essentially the partnership aspect of the study. So not only do we fund the trials, but we also fund the capacity building awards that go either ahead or behind those trials, oftentimes. And then focused on today's conversation, we do have a decent portfolio, again, should be growing in the coming years. In Alzheimer's and related dementias, about 78 million funded so far. And what I'm going to talk to you now about is really how our strategic plan then goes ahead to inform some funding mechanisms that you may think about. So just last year, we released a strategic plan that has 5 ambitious goals, and many of them are reflected in some of the projects that were presented today. So for Dr. Keelar's work, I saw a lot of applicability in the innovation space, in a lot of the advancing the health communications space, all of those aspects. So really increasing innovation in health is one aspect, but then we also have population-focused themes for funding, and as you can see front and center there is promoting the health for older adults. And so that is something that we will be looking at very closely in the coming years. So there are several funding opportunities, and again, you are more than welcome to peruse these. I'm not going to go into large detail, but you can see that compared to our fellow funders, these are not small dollar amounts. They are large, and they are large for a reason, because the idea is that we are at the end of the implementation timeline, right? So once you build your evidence, once you scale your studies, then we want to be the last study before it's implemented out into practice. That's the idea. And it doesn't always happen that way. We certainly have innovative and novel aspects of our funding, and there's some specific targeted awards that do that, and also methodologies behind CER. But that's the idea. And so these are large studies that are impactful. And then the smaller studies are often with engagement awards. So for example, if you do have a community that you're looking to engage with, let's say out in Arizona, you have a population that you really want to think about, how they think about aging, how they think about Alzheimer's disease, for example, you certainly could apply for one of the capacity building or dissemination or stakeholder convening awards. And then you could kind of bring those communities together to help inform your future priorities about research, just as an example. And then, of course, once you conduct the work, we also have implementation award funding opportunities. Some of them are internal, that are limited competition, that are nominated by us as both program officers and the staff, and then others that are external, that also are focused on implementation and dissemination of the work. And this can take a number of different forms. So I just want to invite you to think about us when you're thinking to apply for your work. We have a number of ways to engage, and I'm happy to stay in contact with any of you. There are past funding announcements that we have released on healthy aging, and we may or may not be releasing it again. I'm not at liberty to say yet. But this is our detailed announcement, so stay tuned. We have a board meeting coming up in just over 2 weeks that might have more information for you on this particular focus area. And then there's several free resources for you just on our website to see whether or not you can bring stakeholders together, how best to do that. Even simple things like when you're thinking about participant incentives, what have we found to work and for whom? There's a lot of really nice tools on convening meetings, all of that kind of stuff. So that's all listed here. And then my email and information is here as well if there's anything that you would like to ask or be in touch about. And I think now we can stop for questions. Sorry to jump the gun there.
Thank you. Thank you both. Do we have questions?
We do have an online question that came through. So.
Okay.
But I also just want to thank Dr. Majid. We talked about inviting Pekori here today, and she's like, should I talk about our upcoming calls for grants? And I'm like, yes, please do. So thank you for that.
Sure.
Great, thank you. So the question we have is, the examples you've given of language impairments with PAA seem to be advanced. If that is the case, what do the earliest signs of PPA caused language impairments look like? I work with high IQ, highly educated persons who have self-identified new impairments in their language skills, which are subtle and usually easily masked. And due to their ability to score high on all the traditional cognitive and language screening tools, they are not taken seriously by neurologists.
Yeah, that is true. So we would like to, this is a very important point, that people usually come to neurologists when they very impaired. And at this time, it's really difficult, you know, to do any interventions. The most common problem, I would say, across different subtypes is word finding difficulty. It happens for different reasons, but it happens across subtypes of PPA. So the earlier sign that everyone experiences is a difficulty finding correct word for what they're trying to say. Like, for example, one of the participants said that when this started to happening, she forgot the name of the stove. And that was like one thing that she was trying to explain to her husband, but she couldn't find a word for it. And instead of tomatoes, she would say lomatoes or something like this. So just watching for this progressive difficulty with finding words. Yeah. Yeah, but I think this is really important to find like early point for intervention, because we can really help people when they come in early.
Can I ask a question? So I was just curious about, you found success in this one type of primary aphasia and progressive aphasia. Is there any reason to believe that you would have more success in this particular region because there were more spared areas? It seemed like it was a fairly large region, and maybe there would be more spared areas, and then you'd have more opportunity to get healthy tissue. Or are you predicting differences in the efficacy based on the disease itself?
Because each subtype is associated with different underlying deficit, this specific subtype, we targeted phonological skills. So we thought it would be most beneficial for people who have phonological deficit, because the therapy was targeted at phonology. So in the semantic subtypes, the treatment would look different, because here they have deficit in understanding of language. So we would have to design a treatment that is targeted for that specific deficit. But yes, so
is this MRI guided? I mean,
it is.
Yeah. So yeah, great.
So we would, for semantic, for example, we would select different targets.
Right. Okay.
Yeah.
That's great. Any more questions?
Hello. Congratulations on this. I'm Kathy Runnels. I too am a speech language pathologist, but in another area of practice. Once you discharge your clients, is there any follow-up, or are there things they're supposed to look for or monitor after it's over?
Yeah, we follow up with them. They have 2-month follow-up, so after the intervention is completely done, we follow up with them 2 months after to again test and to assess how they are doing, and also whether they're maintaining the performance. And because in our department there is a clinic, they often get enrolled into the clinic so they can continue intervention. Yeah.
Lee?
Thank you, Annetta. There's such gorgeous data. I saw your grad student present some of them earlier, and it was just stunning. That's such a great question, a follow-on to that. Obviously, this is a progressive aphasia, and the logopenic variant then ends up at the end looking like an Alzheimer's patient. So do you have any idea that how long it lasts? Does it slow the overall progression, or only on the language side? Like, what's your sense of that? Maybe you don't have enough data quite yet to answer that question.
This is a really good question. So we've been administering language tests, but in addition, all other cognitive tests. And people who are receive TDCS with the treatment, behavioral treatment, seems like they don't want to, because the sample is small, but they stable on working episodic memory. So they don't decline as much on episodic memory like the people with the sham group. So I don't, because the group is only 6 in each, so it's pretty small at 12 together, but it seems to be that there is some neuroprotective effect in general, but it's definitely the strongest for the language tasks that we were training. Yeah.
Any more questions? If not, we'll thank the speakers. Thank you.